Hello everyone, I am Bhargav Sawant, an MSc Biotechnology student, and my paper is based on idiopathic pulmonary fibrosis. So, what is IPF? Idio- Idiopathic pulmonary fibrosis is a serious lung disease that destroys the alveolar components and progresses over time without any treatment. There are many unknown causes, and which makes it very difficult to treat. The disease affects a population group over 60 age and has a survival rate of 3 to 5 years. There are over a lakh cases annually. Risk factors include advanced age, male gender, exposure to environmental and occupational hazards, smoking, drinking, and chronic environment insults. The paper, Proteomics in Idiopathic Pulmonary Fibrosis, the quest for biomarkers, was published in Molecular Omics Journal. A little information about the author. Koel Chowdhury is affiliated with School of Medical Science and Technology at IIT Kharagpur, India. Her research interests primarily focus on application of proteomics, genomics, bioinformatics, and helps us to understand the disease mechanisms and identify the biomarkers. Her work involves studying complex diseases like idiopathic pulmonary fibrosis and cancer. She is an expert in various proteomic techniques, including MS, mass spectrometry, gel electrophoresis, and has contributed to advancements in clinical proteomics. Her publications and contributions, she has been authored and co-authored in numerous research papers in different reputed scientific journals. Background of the literature. Definition. Uh, IPF is described as a chronic progressive and fatal lung disease and is characterized or known upon biopsy by the look of scarring of the lung tissue leading to a decline in lung function. Due to the scarring, the patient is unable to respire at the lung's full capacity. The exact cause is still unknown, but it involves complex processes including epithelial cell injury, abrupt wound healing, and excessive deposition of extracellular matrix components. Diagnosis and challenges. Diagnosing IPF is challenging due to its overlap and other interstitial lung diseases. The gold standard diagnosis involves high resolution computed tomography and, in some cases, lung biopsy. Proteomics overview. Proteomics is a large scale study of proteins, including their structures and functions and interactions. It plays a crucial role in understanding disease mechanisms and identifying biomarkers for various diseases, including IPF. Methodology, sample preparation. This involves collecting clinical specimens such as serum, alveoli, tissue samples, or lung biopsies, or lung tissue samples from IPF patients. The tissues are broken down via cell lysis, and proteins are extracted from these samples and then digested using peptides and enzymes like trypsin. Upon this, for separation and identification, gel electrophoresis is performed. 2D gel electrophoresis separates protein based on their isoelectric point and molecular weight. Liquid chromatography is often coupled with MS and helps separating the peptides. Mass spectrometry identifies and quantifies the amount of peptides by measuring their mass to charge ratio. In mass spectrometry, matrix assisted laser ionization is useful for analyzing large biomolecules Electrospray ionization is commonly used for liquid samples and can be coupled with liquid chromatography. Tandem mass spectrometry provides a detailed peptide sequence information, while HPLC, high performance liquid chromatography, is used for separating protein mixtures. Pathophysiology and diagnosis. The pathogenesis, the current understanding of this disease implicates injury to alveolar epithelial cells due to unknown triggers, leading to cascade of responses involving oxidative stress, inflammation, and abnormal tissue repair. Due to abnormal tissue repair, the area around the alveoli gets swollen or has a very systemic differentiation, which accumulates either pus or excess water vapor or moisture, which leads to decrease in the lung capacity. Differential diagnosis. Misdiagnosis is very common due to symptom overlap with other conditions such as COPD and asthma. Timely and accurate diagnosis is essential for um, the disease progression, is essential with reduced disease progression and effective time management. Diagnostic challenges. uh, IPF requires a multidisciplinary approach and is based on clinical, radiological, and histopathological evaluations. High resolution computed tomography is crucial for identifying the usual interstitial pneumonia pattern, a hallmark of IPF. To understand more about this disease progression and to catch the early onset of IPF, certain biomarkers can be identified in our bodies. Current biomarkers are various serum and bronchoalveolar lavage fluids, and they have been identified and isolated, including KL6 surfactant proteins, A and D, IL-8 CXCL13, and these markers include ICAM1 and VEGF. Breath biomarkers. Compounds such as nitric oxide, hydrogen peroxide, and various metals and volatile organic components are also explored as diagnostic and prognostic markers. The study identified several proteins that are differently expressed in IPF patients compared to the healthy control groups. These proteins have potential to serve as a biomarker for diagnosis and prognosis of IPF. The findings provided new insights to pathophysiological mechanisms underlying IPF. The differential expressions proteins are involved in various biological processes such as inflammation, oxidative stress, extracellular matrix remodeling, and immune responses, which are critical in the progression of IPF. Challenges. Pathophysiological insights. The finding provides new insights to pathophysiological mechanisms underlying IPF. The differently expressed proteins are involved in various biological processes such as inflammation, oxidative stress, extracellular matrix remodeling, and immune responses, which are the critical progression of IPF. There are certain challenges in biomarker validation. The study highlights the challenges in validating potential biomarkers, including the need for standardizing the proteomic methods and large-scale clinical studies. It emphasizes the importance of multi-center collaborations to achieve reliable and predictable results. Clinical proteomics. Proteomics involves the study of proteins in terms of their expression, quantification, localization, and interactions. Clinical proteomics applies these techniques to clinical specimens and to understand disease mechanisms and their potential biomarkers. Advances in gel-based and gel-free proteomic techniques have been coupled with mass spectrometry and facilitated high-throughput identification of complex protein mixtures. Bottom-up proteomics analyzes peptides, while top-down proteomics analyzes intact proteins. These approaches are crucial to identify a potential biomarkers for diagnosis and therapy. Future directions. The paper suggested that more research should be conducted on this biomarker validation, including the integration of proteomics with other omics technologies, genomics, transcriptomics, and metabolomics to provide a comprehensive understanding of IPF. It also called for development of robust and validated biomarker panels for clinical use. My understanding, proteomics is a very powerful tool, as the paper suggests. It signifies the role of proteomics in study of IPF. Proteomics enables the comprehensive analysis of protein expansions, interactions, and modifications. The biomarker discovery identifies several proteins that are differently expressed in IPF patients. These proteins have potential as biomarkers for early diagnosis, monitoring disease progression, and assessing treatment response. Technological advancements have been made in proteomic techniques, including MS and LC and HPLC, and are instrumental in identifying and quantifying the amount of protein in complex biological mixtures. Clinical applications. Reliable biomarkers can significantly improve the clinical management of IPF by enabling early diagnosis, personalized treatment, and better monitoring of disease program. Thank you.